Okay, I'm going to explain to you how you can use publicly available databases of DNA sequences in order to um, identify species. And this is part of our first portion of our lab exercise for Lab 6 in genetics. And so the example I'm going to use is one in which someone has told me that they were traveling to an area in South America and saw a interesting lizard that they tried to catch. And all they were able to get was the tail of it. The tail broke off. Many lizards, if you've tried to catch them before, are able to intentionally break off their tail so that they can escape predators. And so this person that was down there telling me about this beautiful lizard they saw collected this tail, brought the tail back with them, and told me that they saw this interesting species, this Sileps paminosus. Okay. But after speaking with them about where they were and what they saw, I thought that they might have been mistaken in their ID of the species because where they were, this species is not known to exist. Okay. And so what I decided to do was take the tail sample that they gave me, extract DNA from it, run a PCR reaction to amplify a very specific gene that I know has been sequenced in other species of lizards. And I'm going to use a publicly available database run by the National Institutes of Health in which all of this DNA sequence data is stored. So there have been millions of publications that have published DNA sequence data, whether it's humans, lizards, frogs, flies, whatever. And whenever those papers are published, the data has to be made available to the community so that the results can be analyzed again by others. And so what I'm going to do is to compare the sequence data that I received when I analyzed the DNA of this tail sample that was brought back to me and compare it to what's out there. And if the species that I have sequenced, this tail tip, has been sequenced before, I will be able to make a match between the tail in my lab and the lizard that my colleagues saw. Okay. So the way I would do this is go to this BLAST website where I can search either proteins or nucleotides. And what I have is a DNA sequence, not a protein sequence. So I'm going to click down here on the left, Nucleotide BLAST, and it comes up with a search menu that allows me to enter the sequence data that I have for this particular species. Okay. And so let me see if it is, let me pull up that sequence data real quickly. Okay, that is not it. Thought I had it quickly. There we go. Okay, so what you see that I've pasted into this box is the DNA sequence I received back from amplifying with PCR and sequencing a specific gene from this lizard tail sample. Okay, I'm going to leave all the other options set to their default settings. So I'm just searching all the databases and looking for similar sequences. So all you do is paste in your sequence that you're interested in here and click BLAST. Now what this is doing is it is comparing the sequence that I'm submitting to all sequences in the database, okay? And it comes back with the results that if I scroll down, here are sequences producing significant alignments or similar sequences. And the details of each of these sequences is listed here in blue on the left. But if you go over to the right, okay, what I see is in this column, the percent of nucleotides that are identical between my sample and the data that is available on this public database. And you'll notice that my tail sample, the sequence that I obtained from that tail sample is 100% identical to a DNA sequence that somebody else has posted from a species, Arthrosaura cockii, okay? Now remember, the person that saw this lizard thought it was this Cylops paminosus, okay? But the similarity is only about 82% identical to a Cylops. It's 100% identical to an Arthrosaura cockii. Here's what an Arthrosaura looks like. You can see they're somewhat similar in that they have a brightly colored tail, but this species is quite different and somewhat distantly related to this one, this Cylops. Okay, the similarity really only being the color of the tail. Okay, so I clicked on that Arthrosaura cockii sequence, and what you see here is the alignment of the Arthrosaura, so subject 1, Arthrosaura cockii sequence that was already in the database, and then my query sequence. So the top here is what I submitted, and the bottom is what is in the database for Arthrosaura cockii. And so you can see here that there are no differences. They match 100%. If I go back, I believe I can go back. There we go. Oops, all right. Let's look at the alignment of the sequence that I submitted lined up next to Cylops paminosus. Okay, and what you see here, look at all these places where there's a missing line between the nucleotide of the query and the subject that was already online, okay? In each of these places, my sample has a nucleotide difference from what's in the database. And so using this database, I can quite quickly compare a sequence that I have in my lab to everything that's been sequenced before and use the similarity to determine what species it is. And this is exactly what you are going to be doing in lab. Okay. Enjoy.